Hello, my name is Chloe, and today I have Amira, Winnie, Lucien, and Margaret with me. Today we are going to present our microbiology project. The topic of our project is Symbiodinium and coral reefs. Okay, Symbiodinium and coral reefs. They actually have a relationship called coral-algae mutualistic relationship. So the Symbiodinium provides oxygen and organic compounds to coral reefs, and the coral reefs in return provide inorganic nutrients and a safe habitat for Symbiodinium. So because the natural environment of coral reefs is very stable, so they are very easily affected by sudden changes of abiotic factors. For example, the increase in sea temperature leads to the death of endosymbiotic Symbiodinium, which results in the bleaching of corals and their eventual demise. So coral reefs are endangered due to many factors, including natural disasters and human activities. Experts state there are only half the amount of coral reefs today compared to the amount 40 years ago. So it is necessary to bring the attention of public back on this topic because the coral reefs are very important regardless to humans or the ecosystem. Symbiodinium and coral reefs are closely related, which means the death of Symbiodinium can lead to the death of coral reefs. So this presentation focuses on the relationship between Symbiodinium and coral reefs and their impact on humans and the planet.
Hi, my name is Amira and I'll be presenting on the Symbiodinium taxonomy. So the genus Symbiodinium consists of 9 broad genetic clades that are genetically highly distinct, which consists of clades A until I. So all clades except E, H, and I have been found in corals, although on the Great Barrier Reef, most corals harbor Symbiodinium types from clade C and to a lesser extent clade D. Clade E is found in Anomalous, and clade F, G, H, and I are common among foraminifera. Each of these clades contains genetically and ecologically distinct subcladal types or strains, and these types can but do not necessarily have to be different Symbiodinium species. Subcladal types often only differ by new nucleotides, and yet they may differ very distinctively in their physiological characteristics, from differences in size and pigment composition to differences in heat stress tolerance. And now I'll be talking about the Symbiodinium structure. So each Symbiodinium cell is coccoid in host bite, meaning it's living in a host cell and surrounded by a membrane that originates from the host cell plasma lemma during phagocytosis. This membrane undergoes a modification to its protein content, which functions to limit or prevent phagolysosome fusion. The vacuole structure containing the symbiont is therefore termed the symbiosome. A single symbiont cell occupies each symbiosome, and it is unclear how this membrane expands to accommodate a dividing symbiont cell. And under normal conditions, symbiont and host cells exchange organic and inorganic molecules that enable the growth and proliferation of both partners. Now I'll be talking about the characteristics of Symbiodinium. So Symbiodinium is a genus of diverse endosymbiotic algae, with genus members commonly referred to as zooxanthellae. So zooxanthellae frequently form symbiotic relationships with a variety of marine animals, including foraminifers and jellyfish. Being algae, organisms of Symbiodinium are autherorhic and eukaryotic, gaining energy from photosynthesis. And during the day, they provide their coral hosts with the organic carbon products of photosynthesis that sustain polymetabolism and growth in oligotrophic waters. And this provision can bother up to 90% of their host's energy needs for metabolism, growth, and reproduction. In return, they receive nutrients, carbon dioxide, and elevated position with access to sunshine. now I'll be explaining about the Symbiodinium growth conditions and role on Earth. So Symbiodinium cells have the best growth rates at normal temperature, which is 258 Celsius, with nitrogen supplies. However, studies indicated that Symbiodinium cells grow well in temperatures above 308 Celsius in nitrogen-free media. So environmental stresses such as elevated seawater temperatures and high irradiance may disrupt the symbiosis, leading to losses of either Symbiodinium cells or the pigmentation of those within the host issues. This is the phenomenon of coral bleaching, and as mentioned before, Symbiodinium provides oxygen and organic compounds as products of photosynthesis to the coral, and in return receives from the host inorganic nutrients and a safe habitat. Hence, they are ecologically important as they sustain the very productive and biologically diverse reef ecosystems worldwide. Now I'll be passing it on to Winnie for its roles on coral reefs.
My name is Wen-Yi. So now moving on to the role, mechanisms, and outcome of the interaction between Symbiodinium and corals. The relationship between corals and Symbiodinium is critical for reef ecosystem sustainability in severely oligotrophic conditions. But it is also the Achilles heel due to its sensitivity to climate change and local stressors. As mentioned by Amira, Symbiodinium establishes obligate relationships with many different animals. But especially for coral reefs, they have been observed to provide up to almost 100% of the coral energy budget by carrying out photosynthesis. Another role Symbiodinium is known for is providing resilience to coral reefs during environmental change. Raised atmospheric partial pressure of carbon dioxide has lowered seawater pH, which hinders reef development, and increased sea surface temperatures, which can cause the coral-algae symbiosis to break down, a process known as coral bleaching. The coral is deprived of symbiont-derived organic carbon during bleaching, which can cause diminished growth, infection, and death. Bleaching episodes have been identified as the main cause of current worldwide coral reef damage, and their frequency and intensity have been rising over the last century. Rare Symbiodinium have been spotted that might be able to increase the resilience of corals, although many do question if the lack of abundance of the rare Symbiodinium might even serve a functional purpose. Next, I'll pass the mic on to Lucien, who will be explaining the mechanism that takes place.
Hello everyone, I am Lucien, and now I'm going to talk about the mechanism that takes place between the relationship of the coral reef and Symbiodiniums. So the process of photosynthesis, metabolic interactions, and calcification are the process that occurs between the relationship of coral reef and Symbiodinium. And as Amira mentioned, the Symbiodinium form a symbiotic relationship with the coral reef because both partners receive benefits from the association via the exchange of nutrients. So the metabolic interactions consist in a system of nutrient recycling between the host and its symbionts, providing them with a competitive advantage in the shallow and low nutrient in the tropical water. So during the photosynthesis, Symbiodinium take in carbon dioxide and water from the air within itself. The water then oxidizes, meaning it loses electrons, while the carbon is reduced, meaning it gains an electron. This transforms the water into oxygen and carbon dioxide into glucose. And the Symbiodinium then releases the oxygen back into the water and stores energy within the glucose molecules. So now move on to the metabolic interactions and calcification process. So in coral reef, photosynthesis and calcification do not occur independently. So the Symbiodinium provide the coral with photosynthetic products such as glucose, glycerol, and amino acids. So these products are used by the coral reef to manufacture proteins, lipids, and carbohydrates, as well as the calcium carbonate to carry out the calcification. So the Symbiodinium also remove phosphate that inhibit calcification and thus enhance crystallization of the calcium carbonate. So the Symbiodinium acquires metabolic breakdowns and nutrients from the host, such as inorganic carbon and nitrogen, phosphate, and the other inorganic compounds to produce enough organic molecules to fulfill up to 95% of the host energy demand. And moreover, carbon dioxide uptake necessitates the proton pumping adenosine triphosphatase to acidify the boundary layer and lead to the formation of bicarbonate or HCO3, which is then quickly and externally dehydrated into the carbon dioxide by the membrane-bound carbonic anhydrase, where it is converted into the HCO3 by the cytosolic carbonic anhydrase, preventing the carbon dioxide leakage. And lastly, dissolved inorganic carbon is then supplied to the calcification site.
Now I'll be continuing on with the outcome of interaction. Symbiodiniums are one of the most important organisms that live in symbiosis with coral reefs. They are responsible for providing 90% of the nutrients for corals and the unique and beautiful colors of many stone corals. Although corals do not perform photosynthesis, due to their close relationship with symbiodiniums, they frequently react to their surroundings in the same way that plants do. Coral reefs require clean water for photosynthesis so that sunlight can reach the algae cells. As a result, they are only found in waterways with a low concentration of suspended particles or in water with low turbidity and low productivity. In these environments, the interaction between the algae and coral polyp permits a tight recycling of nutrients. The relationship between Symbiodinium and their hosts determines not only the rate of coral reef growth, also known as the calcium carbonate deposition, which was explained by Lucien, but also how the system responds to environmental stress. Symbiodinium, in turn, benefits from the host protected microenvironment, which includes access to light, inorganic nutrients, and dissolved inorganic carbon. Without this partnership, coral reefs would not be the prolific ecosystems we see and rely on today. I'll be passing the mic on to Margaret, who will be continuing with the impact of changes on humans and the planet.
Hi, my name is Margaret, and now I'm going to start off by presenting the impact of coral reefs to humans. So first of all, coral reefs will serve as a protection from storm and wave action. Coral reefs serves as buffer and protection from the pounding of ocean waves. Many beaches and coastal cities would be in grave danger to storm damage and wave action in the absence of coral reefs. In the next slide, I will give a statistical example on how coral reefs really impact us humans from the protection of storm and wave action. Here is a statistical example. In Maldives, almost 10 million USD is used to repair the damage caused by storm and wave action when coral reefs and sand were mined away. This shows that we have to use this amount of money to repair the damage caused by natural phenomena in the absence of coral reefs. Now I'm going to present about the impacts of coral reefs to the planet. Firstly, coral reefs help fish to boost reproductive success. In a research done by Metabolic SL, Brentford AN, Gadani P et al. In 2022, they found that noise pollution can cause stress, distraction, height injuries, and interrupt all levels of marine wildlife. Hence, we are advised to avoid driving near coral reefs to enhance fish fitness. In the next slide, I will provide a statistical example from the research that they have done. Secondly, Symbiodinium are essential photosynthetic symbionts in corals. Symbiodinium aids in 90% of carbon's fixation via photosynthesis to meet the energetic needs of the coral host. Basically, Symbiodinium help coral reefs to carry out the majority of their photosynthesis process to provide energy for the coral reefs. Here is the statistical example provided by the researchers in the previous slide. So as you can see here, they provided two types of charts. Here is a bar chart, while B is a line graph. So first of all, let's see this bar chart A here. In the x-axis here, we have the busy boating for red and limited boating or no boating in blue. while in the y-axis here is the number of nests. So as you can see here in the busy boating area, this red area over here, the number of nests is 40, while in B is around 45. So as you can see here, even though the difference for the total number of nests is like only 5, but in this striated column over here, you can see that it means lost all offspring. You can see that in this busy boating area, the number of nests where they lost all offspring is higher than in the limited boating area. So this shows that in the busy boating area, the chances for the fishes to lose all their offspring is higher than in the limited or no boating area. Moving on to this line graph over here in B, in the x-axis here we have time in terms of days, and y-axis is the survival probability. As you can see here, in this red line over here, which means the busy boating, it is more steeper compared to the limited boating or no boating. So which means since it is steeper, it means that the survival probability for in the area of busy boating is lower compared to the limited boating or no boating area. So in this statistical example shown, the noise pollution really affects the fish fitness and in the coral reefs. So here is our references, and I hope our presentation was enough entertaining for everyone. Thank you for listening, and this is Margaret signing off. Bye bye.